Hello and welcome to Erlangen in Germany. My name is Ricardo Grieshaber-Bouyer. I'm a professor of Clinical Systems Immunology at FAU and head of the Clinical Trial Unit at the Department of Medicine 3. You might have heard of Erlangen recently. It's one of the hottest places for immunology, clinical care and research right now across the world. It was the first place for a patient with autoimmune disease to receive CAR T-cell therapy, and also the first place in the world for a patient with autoimmune disease to receive T-cell engager therapy. And we're very excited about all this progress. A little bit about my background. So I studied medicine and business administration and then trained in internal medicine, rheumatology and immunology. Why did I choose this specialty? Well, quite simple. It's the most exciting field in medicine. We get to see a lot of very complex systemic immune-mediated diseases, patients that have a very high unmet medical need, despite the breakthroughs that have already been made in this space. And there remains a high need for understanding the molecular pathophysiology of these diseases and then designing better targeted therapies and then also deploying them for patients. So we are a very large team. The entire department has over 200 employees. Many work in research labs, and also our clinical trial unit is a big unit, has over 30 employees, over 10 physicians. And we all work on innovative clinical trials to bring innovative medicines to patients with high unmet need. I think it's important to get this behind the scenes view, because really now is a very transformative and very exciting time for medicine and especially for immunology, because we are starting to think in a different way how we can potentially treat autoimmune diseases, not just chronically, but really to reset the disease, leading to a state of a long-lasting drug-free remission, ideally where we have eradicated or wiped out the autoreactivity that is underlying the disease and have achieved a state for the patient where they can be disease-free. A couple of years ago, this was not really thinkable, but in the past 4 years, really, we have learned so much about how for instance, redirecting T cells against target cells can be a more efficacious way of eradicating them. And this has led to the idea that the autoimmune disease can really be reset and potentially cured. So rheumatic diseases have a very high disease burden in the population. Over 3% of the population live with autoimmune and rheumatic diseases. And rheumatoid arthritis is one of the most common autoimmune diseases in the population. And Sjogren syndrome is also very common. And the issue with rheumatic diseases right now is that even though we have therapies that can alleviate symptoms, such as TNF blockade, for instance, which has changed the way how autoimmune diseases have been treated, they rarely ever achieve a state of a patient being in a drug-free remission. So the current standard is chronic immunosuppressive therapy. And even when patients are in remission, they can continue to accumulate organ damage and ultimately loss of function. And what patients are echoing to us over and over again is that they don't want to be taking any medication. So this is really the number one priority of the patient. They want to be disease-free, but also medication-free. And so what this means to us is that we need new innovative therapies that are able to attack the underlying mechanistic issues that drive autoimmune disease pathology, not just suppress certain signaling pathways in a chronic manner, but really correct the underlying defects of the immune system and thereby grant a state of a drug-free remission. That is the vision and the goal to manage immune-mediated diseases in the future. So from a fundamental perspective, the goal is to deplete a certain cell type in the most efficacious way possible. And there's different approaches to target cells, for instance, using canonical monoclonal antibodies. But really, T-cell redirecting therapies are showing to be much more powerful to target and very deeply deplete target cells. The CAR T-cell therapy and the T-cell engager therapy mechanistically have a lot in common, right? And so the difference is just how do you get there? So what a T-cell engager does is it leverages all the T-cells that are already present in the body and allows them to recognize the target cells expressing a very specifically chosen surface antigen. And it acts as a bridge between the T-cell receptor on the T-cell side and the target cell expressing that target antigen. And then by engaging the T-cell, it becomes activated. It releases perforins and granzymes through a cytolytic synapse. And this leads to the depletion of the target cells. The manufacturing is very scalable for protein-based therapeutics because this has been done for many decades. And so it's possible to reach a very high throughput and manufacture large batches that can then be stored and readily administered to a patient when they become eligible and when the decision is made to use a T-cell engager. So this is a great advantage of this treatment approach compared to many other approaches where, for instance, individualized manufacturing might delay the treatment time, but here you can go from making a decision to dosing pretty much right away. And for the CAR T-cell therapy, the T-cells are genetically engineered to express a chimeric antigen receptor, which allows them to recognize the target cells. And this currently means giving the patient lymphodepleting conditioning chemotherapy to make space for the CAR T cells and then giving these cells back as an infusion. As is currently the case, a cellular therapy is given in an inpatient setting due to the preconditioning therapy, and then also to monitor the patient during the expansion phase to be able to detect and then treat potential side effects very quickly. For the T-cell engagers, the logistical part does not require hospitalization. Right now, in these early clinical trials, TCEs are given as inpatients for safety reasons to be able again to monitor patients. But then ultimately, once the safety profile becomes more firmly established in a safe therapy scenario, these therapies can be given as outpatients. So what I can share from our own experience, which now covers way beyond 40 patients with autoimmune diseases, we haven't seen any higher grade cytokine release syndromes or neurotoxicity syndromes, and the safety profile appears to be pretty good. The treatment can start pretty much right away because nothing has to get produced for the patient. So once we make the decision that this trial is the right approach for them, we just have to complete screening examinations that can be completed in a day. And then after the sponsor approves the patient's eligibility, treatment can commence right away. And so there's a very short lag time, only usually of a very few days to be able to start treatment. And I think this is also something that makes T-cell engagers quite compelling for the future, because if a disease is active and there's an indication to switch treatment or to escalate treatment, then there is no harmful delay and the patient can get treated right away. As we had seen in 2021, CAR T-cell therapy provided a really strong clinical response in refractory autoimmune disease. And ultimately, we thought, why not use a different approach that is more accessible off the shelf to deplete B cells in autoimmune diseases, and hence the idea of using T-cell engagers. And that's how all of this work got started here.
I'm Laura Bucci, a rheumatologist, an Italian rheumatologist working in Erlangen. I decided to join this group and this lab to really be involved in understanding the mechanism behind autoimmune diseases. To me, what's really fascinating about T-cell engagers is that this is a really good alternative to reach a deep result in patients with autoimmune disease. That's really one aim that it's difficult to achieve, because we think about diseases in general always referring to elder population, but actually people really experience that. People who still have a life and want to have kids and want to have, a normal life. So if we can help them reaching that status, I think this is our aim and our win. So we know that basically the T-cell engagers are engaging the T-cells against the B-cells. There are for sure different mechanisms. In this lab, we are approaching this different mechanism by studying what happens in the blood of the patients and also in the tissue of the patients. So an in vivo approach, and we are trying to establish also the in vitro approach to stimulate the cells with different concentrations of T-cell engagers and to analyze not only the B-cell depletion, but also the environment and how the other cells act.
So a phase 1 trial is really primarily a safety trial. So the purpose of the trial is to identify a dose that is well tolerated in patients and start to understand pharmacodynamic implications of this dose. For instance, the successful mechanism of action, in this case, the depletion of B cells in the blood and in the tissues. We know now that many immune cells don't live in the blood, but actually live in tissues. And since the tissue is the place of action, it's important to study the tissue to understand in active autoimmune disease how, for instance, T cells and B cells are interplaying in inflamed tissues, and then also to study longitudinally which tissue factors are relevant for the successful depletion of pathogenic cells from tissue, and also leveraging longitudinal tissue biopsies to confirm the treatment success on the tissue level. And we will learn this through the phase 1 trial. Also, what's very unique about the phase 1 trial design is that patients don't get treated in parallel, especially at the beginning, but rather there is an observation period after one patient receives their dose to make sure that no side effects occur that would prompt us to stop the trial. And only when this window is closed, then the next patient can receive their dose. And then also the other thing that is very characteristic about such a study is that we review the data on a regular, ongoing basis and think about how to escalate the dose in the safest way possible without exposing patients to an increased risk. And so we can, for instance, decide to elevate the dose in the next cohort, or we can decide to enroll additional patients in the present dose cohort, all depending on the clinical data as it emerges. In patients with rheumatoid arthritis, but also lupus, Sjögren's disease, and other indications, we will learn, for instance, how the pre-medication is taken up by the patients, what the safety profile of the therapy is like. We will continue to learn about the logistical consequences of the treatment, and we will see which aspects of the current treatment protocols are really necessary and which features can get more relaxed in the future. So far, everything that we have seen, both coming from cellular therapies and from T-cell engagers, is that these treatments have a better safety profile than when they are used in hemato-oncology. And I think now with T-cell engagers emerging that are specifically developed for autoimmune disease, the safety profile will continue to get better and better. And for the referring providers, this is a good relationship also, that they know that they can refer their severely affected patients to us and get access to good treatment options that are being evaluated now in clinical trials. So as the study is rapidly expanding, the number of sites will continue to grow, and up-to-date information on which closest site is available can be found online for instance, by checking on clinicaltrials.gov or on the website of the study sponsor for an up-to-date list of participating centers.
Now that we've explored the science behind T-cell engagers, let's examine what this means for patients in practice. Dr. Grieshaber-Bouyer brings us inside his clinic where he meets with a patient considering participation in a clinical study with a T-cell engager.
If I decide to go forward with this study, what would the treatment day look like?
Yes, I'm happy to walk you through it. So on the treatment day, we will already have completed your eligibility criteria, and you will arrive in the morning. We will draw bloods again just to make sure that the renal function is stable, that you don't have any elevated liver function tests, that there are sufficient B cells in the blood, that there is no low platelets, for instance, or no other cytopenias, enough white cell counts in the blood. Once these lab tests are confirmed, we will give you pre-medication. For that, we will typically equip you with 1 or 2 peripheral lines. We will give you pre-medication that usually consists of glucocorticoids, so steroids, Tylenol to prevent fever, and then an antihistamine. These medications can make you a little bit drowsy. And then after 1 to 2 hours, we will commence treatment. It's a single subcutaneous injection. It's prepared fresh from the pharmacy on the same day. You will receive the subcutaneous injection, and at that point, really we just monitor you for any potential side effects. So we look at the blood oxygen saturation, we look at the heart rate, we look at the blood pressure, we regularly take your temperature, and we monitor you for any symptoms such as confusion, other neurological deficits. We will do twice a day with you a very simple score where we look at your orientation and your ability to perform normally on a neurological scale. And then really after the observation period is completed, after 48 hours to 72 hours, then you are discharged. And we continue with the next dosing, which, for instance, in the second dose cohort is on day 8. But this is, of course, dependent on the study design.
Then we asked patients what they wanted to know about clinical studies with T-cell engagers.
Hi, I'm Amber. I have moderate lupus. I'm currently on a treatment plan for lupus that works okay for me, but I'm considering participating in a clinical trial. So, how do I weigh the risks and benefits of leaving a treatment plan that's actually helping, even if it's just a little bit, to join a clinical trial?
So I would say this really depends on the clinical trial. If you are stable on your current medication, it's probably not the right situation to go off this medication and to join the clinical trial of an experimental drug. Such a switch might come later, more naturally, in later phases of clinical development or after a compound is approved. At this point, early clinical trials are mostly meant for patients that experience inadequate response to standard of care and have active disease.
If I were to join a trial like this, what would the experience look like? Would I need to be hospitalized? How often would I get treatment? And is that something I would have to do more than once?
So in the current stage of clinical development, T-cell engagers are administered in an inpatient setting, meaning that there's a short period, usually 24 to 72 hours, where the patient is hospitalized for monitoring and for safety reasons. And in the foreseeable future, this will likely become more relaxed as T-cell engagers will become applied in an outpatient setting. In terms of the dosing schedule, most studies right now are set up in a way that there is only an initial treatment phase, followed by a phase in which no chronic therapy is administered. And the studies will then show whether this leads to a long-lasting state of drug-free remission or if redosing is necessary in the future.
And is there a possibility that the study medication could get me into a treatment-free remission?
So while we don't have any long-term data yet, how long and how durable the responses to T-cell engagers are, data is emerging that some patients experience long-term responses, and therefore the hope for the future is that this will allow patients to experience extended periods of a drug-free response.
If this treatment shows promise, how would someone like me get access to it? And what kind of patients are likely to benefit the most from this kind of therapy?
So patients that are most likely to benefit, this depends a little bit on the target that is pursued by the T-cell engager. In the context of targeting B cells via CD19 antigen or also via BCMA antigen or other B cell antigens, this is mostly patients that have autoantibody mediated and B cell mediated autoimmune diseases. And this is actually a very large group of patients with autoimmune diseases that encompasses, for instance, rheumatoid arthritis, systemic lupus, inflammatory myositis, Sjogren syndrome, systemic sclerosis, and many other types of immune-mediated diseases. And the way to get access to this treatment right now is only via clinical trials. And ultimately, once it is approved, this will be prescribed by the rheumatologist and immunologist.
Can you maybe explain what a phase 1 study is? Also, what are you doing to ensure my safety during the study?
So a phase 1 study is explicitly designed to be a safety study. The purpose of the study is therefore to expose patients to the least risk that is possible, whilst safely increasing the dose to reach more efficacious doses. And typical characteristics of a phase 1 study are a very small, limited number of patients that can receive the treatment. Also that patients are typically dosed sequentially and not in parallel until each dosed patient has been evaluated for potential side effects. And there is also very frequent evaluations of the investigators and the entire study group reviewing the data on an ongoing basis to determine, for instance, if the dose can be increased or if the dose has to be maintained and if additional patients have to be enrolled in the current dose level.
My name is Cristina Montoya. I'm a woman living with Sjogren's disease and rheumatoid arthritis. And my first question to you is, how is this treatment different from what's currently available to people living with autoimmune diseases like Sjogren's?
So currently, no treatment is available that is similar to a T-cell engager therapy. T-cell engagers target a specific type of immune cell. In the case of the currently studied T-cell engagers, they recognize B cells. And the goal is therefore to deplete B cells in a very efficacious way. And current approaches to deplete B cells are limited by their ability to deplete cells in the tissues. And therefore T-cell redirecting therapies are uniquely poised to lead to a deeper depletion of such cells in the tissues, potentially accomplishing immune reset.
Sjogren's has historically borrowed medications from other diseases, and understanding that CLN-978 is also being studied in lupus and rheumatoid arthritis, is this really a potential treatment for Sjogren's?
So it has many reasons why no treatments have been approved specifically for Sjogren's disease so far. But it would not be right to think that treatments that are also approved for other disease indications don't make sense in Sjogren's disease. In fact, Sjogren's disease is very much so a B-cell driven disease, and therefore targeting B cells therapeutically, mechanistically makes sense in Sjogren's just as it does in other autoimmune diseases.
I'm Shelley Fritz. I live with rheumatoid arthritis. And if I wanted to explore this clinical trial, how would I go about finding out if I'm eligible?
Yeah, so the eligibility ultimately is always confirmed by the enrolling physician using a standard set of inclusion and exclusion criteria. And then once these inclusion-exclusion criteria are followed, a patient can be enrolled in a trial.
If T-cell engagers prove to be effective, would patients need to use T-cell engagers in combination with other therapies to control their RA symptoms?
So it's conceivable to use T-cell engagers in combination with other therapies. but so far there is no data that would show that this actually needs to be done. And so right now the goal of clinical development is to find out if T-cell engagers can be efficacious enough and even more efficacious than current treatments without having to combine them with other therapeutic approaches.
How long do you think that the clinical development process could take from this phase 1b stage to getting to regulatory approval?
So currently T-cell engagers are in phase 1 trials, and it is unlikely that in the next year, in 2026, T-cell engagers are approved for treatment. However, we could see the opening of registrational trials potentially as early as the beginning of 2027. So from my perspective as a clinician and scientist, what really excites me to see is that it took overall less than 1 year from us first publishing our findings on T-cell engagers in autoimmune disease in Nature Medicine and then in the New England Journal of Medicine to launching the first trial of a T-cell engager in autoimmune diseases. And it's just great to see all this work come full circle from the initial discovery into the early clinical development and now learning many additional mechanistic things about their activity in patients. And so this is all, of course, ongoing work. There are many updates of data and results to come, so definitely stay tuned for more results that we will present soon at conferences and publish in journals, and reach out if there are any questions and also if there are patients that you would like to refer to the studies. Okay, well, I hope this presentation was exciting for you. Stay tuned for many more updates to come as we release new data from our clinical trials and from our research studies. And thank you for watching.